डर लग रहा है कैसे करें रिवाइज बायोकेमिस्ट्री, इतना सारा है, टाइम कम है. सो इन दिस सीरीज क्रिस्प रिव्यू बायोकेमिस्ट्री बाय पूनम अग्रवाल मैं आपसे डिस्कस करूंगी कि बायोकेमिस्ट्री को कैसे रिवाइज करें शॉर्ट टाइम में. ये फर्स्ट वीडियो है विच इज़ कार्बोहाइड्रेट मेटाबॉलिज्म पार्ट 1, जिसमें मैं सारे कार्बोहाइड्रेट मेटाबॉलिज्म की बात करूंगी एक्सेप्ट ग्लाइकोजन मेटाबॉलिज्म. और आप ये देखोगे कि थोड़े ही समय में आप कैसे इस टॉपिक को रिवाइज करते हो. और सबसे पहले हम बात करेंगे ग्लाइकोलिसिस की, जो मेन स्केलेटन पाथवे है. ग्लाइकोलिसिस का पर्पस है एटीपी प्रोडक्शन, और ये होता है साइटोसोल में इन ऑल द सेल ऑफ द बॉडी. Whether it is aerobic or anaerobic condition, you have glycolysis going on, and the main purpose is ATP production. But in RBC, you also have formation of 2,3-BPG in a shunt of this glycolytic pathway, which is called Rappaport-Lubering shunt, RL shunt, Rappaport-Lubering shunt. Right? So the glycolysis is a very vital pathway, and we're going to see the core aspect of it. In this, the glucose is converted to glucose 6-phosphate by glucokinase or hexokinase enzyme, and then glucose 6-phosphate is converted to fructose 6-phosphate by phosphohexose isomerase enzyme. This is phosphofructokinase 1 enzyme, and this fructose 1,6-phosphate is going to break into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate in the next step, catalyzed by aldolase, which may be either A, B or C type. Now at this step, next step, inorganic phosphate will be required, and glyceraldehyde 3-phosphate dehydrogenase enzyme will convert this glyceraldehyde 3-phosphate to 1,3-BPG. This 1,3-BPG is converted to 3-PG by PG kinase enzyme, phosphoglycerate kinase enzyme. This is going to be the mutase enzyme, and this is going to be enolase enzyme, which is going to dehydrate 2-phosphoglycerate to form you phosphoenol pyruvate. Then pyruvate kinase will be required, and then this conversion is going to be a spontaneous process. So there are a number of steps. There are total 4 kinases: glucokinase, hexokinase, PFK, PG kinase and pyruvate kinase. Out of these 4 kinases, 3 kinases are irreversible: glucokinase, hexokinase step, PFK1 step, and which one? Pyruvate kinase step, right? In other than these 3 steps in the glycolysis, all other steps are going to be reversible. This is important for you to know the reversibility, irreversibility of glycolysis, because you will see shortly that many of the steps we are going to use in the gluconeogenesis. So now you have seen how the glycolysis is taking place. Glucose is converted to enolic pyruvate, then ketotic pyruvate. Then comes the next aspect of the glycolysis, that what is the future of this pyruvate, which is produced as an end product of aerobic glycolysis. So for that, you must know you have one enzyme called lactate dehydrogenase, which has to act in anaerobic condition to convert this pyruvate to lactate, again in a reversible fashion, right? But in aerobic condition, the future of this pyruvate is entry into the mitochondria matrix, and there this PDH complex is going to convert this pyruvate to acetyl-CoA, which is further utilized in the TCA cycle. So this is how the glycolysis is taking place, and the pyruvate is either converted to lactate in an aerobic condition, or the pyruvate is having the future in PDH complex in aerobic condition, right? So now when we talk about ATP consumption, there are 2 ATP which is consumed, one at GK step, another as to PFK1 step. And then you have 2 ATP production, one is at PG kinase step, another is pyruvate kinase step. This is called substrate level phosphorylation, because you don't need electron transport chain NADH, FADH2 for ATP generation. This is a very vital step which is producing you ATP without any need of oxygen, without any need of mitochondria, etc. So this is called substrate level phosphorylation, which is there at 2 places in glycolysis. One is PG kinase step, another is pyruvate kinase step. You must know that this glyceraldehyde 3-phosphate onward, these reactions are going to occur twice in this glycolysis. Glyceraldehyde 3-phosphate downward, what all reactions you are seeing, all these reactions are occurring twice because this DHAP is converted in a reversible fashion by phosphotriose isomerase to glyceraldehyde 3-phosphate, right? So you must know all this glycolysis steps. And you see the glyceraldehyde 3-phosphate dehydrogenase step, you have formation of NADH because NAD is the coenzyme of this enzyme. So if you talk about the gain in this pathway, you have 2 NADH generated here, 2 ATP here and 2 ATP here. I am telling 2 because these steps are going to occur twice, once for original glyceraldehyde 3-phosphate and next time for DHAP, right? So we have production of 2 NADH here, 2 ATP here and 2 ATP here. This gives you 4 ATP and 2 NADH. This is the gain. This is going to produce you 5 ATP in best of circumstances when malate aspartate shuttle is being used, and this 4 ATP is 4 ATP. So you get to have plus 9 ATP in the glycolysis. But if you have noticed, we have utilization of 2 ATP at the initial steps of glycolysis. So for net ATP generation, we have only 7 ATP being formed because 2 ATP is utilized at glucokinase and PFK1 step. This I am telling you when NADH is capable of making you ATP, and that is possible only in aerobic condition when oxygen and mitochondria etc both are present in the cell. Suppose if either the oxygen or the mitochondria is absent, that time this NADH is utilized at LDH step, right? The NADH which is coming from top from glyceraldehyde 3-phosphate dehydrogenase step is actually getting utilized here to convert, to reduce the pyruvate to lactate, and this way the NAD is reclaimed, which will be running the glycolysis continuously. So we will not gain this 5 ATP in anaerobic media, and we are left only with these figures, I mean only with 4 ATP in anaerobic condition. Anaerobic condition you have only total 4 ATP, and that too by substrate level phosphorylation which we got, and net ATP generation is only going to be 2 ATP. This ATP statistics is very, very important in glycolysis, and you must know this, right? So this is about the route of the glycolysis, and I've told you the reversibility, irreversibility. Now coming to the next aspect of it, we always wonder that why glucokinase, why hexokinase, is there any difference between these two enzymes, name and distribution and their functionality? So yes, of course, they are actually the isoenzyme. The glucokinase is nothing but it's like type 4 isoenzyme of hexokinase, but it has got a very important characteristic which is unique to this glucokinase. So that is why this new name glucokinase is given to this glucokinase enzyme. So this table is very important table which is in nutshell talking to you about the difference of hexokinase and glucokinase. As I have told you, it is type 4 isoenzyme. Glucokinase is type 4 isoenzyme of hexokinase, and when we say hexokinase, we simply mean to say that we are talking about various isoenzymes like type 1, type 2, type 3 isoenzymes of hexokinase. You must know, now I am just reading all these differences one by one, try to understand this. When we talk about glucokinase, it's distributed in beta cell, pancreas and liver, and all other cell we have various other isoenzymes of hexokinase. And when we are talking about glucokinase, it's a specific for glucose, but hexokinase is, as the name implies, it is non-specific. It can act on fructose, galactose, even on glucose, right? The hexokinase is having high affinity to glucose, so it is having low Km value, and glucokinase is having low affinity, so it is having high Km value. You know what is Km value? Km value is the amount of substrate which is needed to get the half max velocity. So you need very low amount of hexokinase to get half max velocity, by which glucose will be converted to glucose 6-phosphate. So practically speaking, if Km value for hexokinase is just less than 1 milligram per deciliter, so you understand practically hexokinase is going to be active at all the glucose scenarios, whether it is 40 milligram per deciliter or 100 milligram per deciliter. Hexokinase is going to be active at all the time, irrespective of blood glucose level, and it will be converting the glucose to glucose 6-phosphate. But pay attention to the hexokinase. It is requiring very high amount of glucose, very high amount of glucose, 180 milligram per deciliter, which is only seen transiently postprandially. This high amount of glucose will be required to get the half max velocity if the glucokinase wish to convert that glucose to glucose 6-phosphate, just because glucokinase is having low affinity. So that gives a very practical aspect to it, that glucokinase, which is there in the liver and beta cell, will be practically active only when very high content of glucose is there in the body fluids, and that is seen in well-fed state. And hexokinase is going to be active all the time, right? And when we talk about induction, the hexokinase is not inducible, but glucokinase is inducible in presence of insulin. And hexokinase is having inhibition by glucose 6-phosphate, which is its product, but glucokinase is having no such inhibition. And insulin, as I said, hexokinase is not synthesized in excess quantity, neither its existing enzyme activity is going to increase. I mean to say, insulin is having no effect on hexokinase, but insulin is going to have impact on the glucokinase, whereby it is going to increase the synthesis as well as the activity of the glucokinase enzyme, right? So this was regarding the difference between glucokinase and hexokinase. Now the rate limiting enzyme for glycolysis is PFK1, rate limiting enzyme. This rate limiting enzyme is allosteric enzyme, which is having its positive and negative allosteric factor. So if you see this picture is talking to you about the the positive and negative allosteric factor for PFK1 enzyme. Fructose 2,6-bisphosphate, AMP, they are going to be a positive factor for PFK1, which are going to stimulate the activity of PFK1 and upregulate the glycolysis. And negative factors are going to be ATP, citrate, and proton. So logically also everything can be explained, but I'm not going in detail of everything. But you can understand the energy abundant state will not be needing the glycolysis. So PFK1 is inhibited, and energy deficient state will be upregulating the glycolysis to compensate for ATP. What I just wanted to tell you here, that this fructose 2,6-bisphosphate is the key compound which is regulating the PFK1 activity, and its level is going to increase in presence of insulin. Insulin increases this level. So insulin increases this level, so PFK1 activity is also increased. On the other hand, the hypoglycemic hormone glucagon is going to decrease its level in the cytosol. So in hypoglycemia, when the person is not eating anything, that time liver would not like to go for the glycolysis, because glucose level is so low in the blood, and anyways the glucokinase is not going to be that happy and that active. So in presence of hypoglycemia, the glucagon will be secreted, and this glucagon will have inhibitory impact on the ongoing glycolysis in the liver. And this is all mediated via fructose 2,6-bisphosphate level. So insulin is going to increase the level, and glucagon is going to decrease the level of fructose 2,6-bisphosphate. I'm not going in detail of alteration of fructose 2,6-bisphosphate level in presence of insulin and glucagon. For learning that, you have to see my other videos, right? So this is regarding the PFK1, which is the rate limiting enzyme. Please understand, PFK1 is a different enzyme, and PFK2 is a different enzyme. PFK2 is the enzyme which is playing role in the regulation of not only glycolysis but also of gluconeogenesis. But PFK1 is the exclusive enzyme for glycolysis. I hope it is clear to you. So this is brief regarding the glucose glycolysis. And I think I must proceed ahead, and because I need to cover a lot many things, so all detail I cannot discuss in this short video for a particular pathway, but certainly I'll give you the overview of carbohydrate metabolism, and you will see by the end of the video that how beautifully galactose, fructose, gluconeogenesis, everything is linked to this glycolysis, right? Now coming to the PDH complex, which is my next topic of discussion, I said in the mitochondria matrix, this pyruvate will be going in aerobic condition where it will be oxidatively decarboxylated to produce your acyl-CoE. And in fact, it's a very, very complex process where number of enzymes and coenzymes will be required. You can visualize this picture, and this is the PDH complex picture. These are the three enzymes E1, E2 and E3 in the PDH complex. They are pyruvate dehydrogenase, dihydrolipoyl transacetylase, dihydrolipoyl dehydrogenase. If you want to locate the pyruvate, this is the pyruvate, and this is where is the acyl CoA, right? So you have to search for the acid. Yeah, this is the acetyl CoA. So you see pyruvate is going, it's like converted to acyl CoA, and there will be loss of one carbon dioxide. To facilitate this reaction, you need to have a whole complex of enzyme which is called PDH complex enzyme. This is having three enzymes as I have illustrated here with highlighter, and you have 5 coenzymes, 5 coenzymes है. They are thiamine, ठीक है, lipoic acid, coenzyme A, FAT and NAD. इस तरीके से you have 5 coenzymes in the PDH complex. Ultimately, the reducing equivalent is being accepted by NAD, so we gain NADH at PDH complex reaction. So it's a happy news that we get NADH also at PDH complex reaction. For one pyruvate, one NADH, for 2 pyruvate, 2 NADH we're going to get, right? And this comes the TCA cycle. TCA cycle is there in the mitochondria matrix. See, there only the pyruvate is oxidatively decarboxylated, giving you acyl CoA. So you see how the TCA reactions are going to take place in the mitochondria matrix, and you have got number of enzymes in the TCA cycle which are distributed predominantly in the mitochondrial matrix, except one enzyme which is there on the inner membrane, which I'll be telling you. So this is going to be the citrate synthase enzyme which is catalyzing irreversible equation. Then this is going to be aconitase enzyme. Then this is also, you know, the aconitase, because intermediate is cis-aconitate. This is isocitrate dehydrogenase. Then this is importantly alpha ketoglutarate dehydrogenase complex, just like PDH complex. This also is having 3 enzyme and 5 coenzyme. This is succinate thiokinase, which is catalyzing substrate level phosphorylation, generating GTP in the liver and other cells, the ATP. Then you have succinate dehydrogenase enzyme which is generating FADH2. You have fumarase enzyme which is using water as a substrate. You have malate dehydrogenase enzyme which is producing NADH. NADH is produced at 2 more places. One is isocitrate dehydrogenase, right, and the alpha ketoglutarate dehydrogenase complex. So how many NADH you have received? 1, 2, 3. 3 NADH and 1 FADH2 and 1 GTP. So if you are going to calculate the energy which you are going to gain from one run of TCA cycle after complete oxidation of one molecule of acetyl CoA, you will get 3 NADH, 1 FADH2 and 1 GTP, which is equivalent of 1 ATP, right? कितनी आई ATP? 1.5. This is going to be 7.5. 10 ATP मिल गए. So 1 TCA cycle you are going to gain 10 ATP. So 1 glucose, 2 pyruvate, 2 acetyl CoA, 20 ATP. So if you see, I always tell that, remember, 9 plus 5 plus 20 is the total number of ATP after complete glucose oxidation in aerobic condition. 9 comes from glycolysis, 5 comes from PDH complex, 20 comes from run of TCA cycle where 2 acetyl CoA is getting completely utilized. This is giving you the figure of 34. So total ATP generated after complete aerobic glucose oxidation is 34, and for the net number you just have to deduct 2 ATP from the glycolysis. Sorry, that is going to be 7 ATP net ATP from the glycolysis. 5 PDH complex को नहीं छेड़ना है, that will be there, and TCA को भी नहीं छेड़ना. This is going to be 32 ATP net after complete net ATP production after complete aerobic glucose oxidation. This number you must remember. You must know the difference between the glycolysis and glucose oxidation. As far as the aerobic condition is concerned, the glycolysis is just a part of aerobic glucose oxidation. But yes, of course, in anaerobic condition, the anaerobic glucose oxidation is synonym of anaerobic glycolysis, because we just have formation of glucose to lactate, right? After that, beyond that, there is no PDH complex, there is no TCA. So whenever we are talking about anaerobic glucose oxidation, it's synonym of anaerobic glycolysis, which is going to give you total 4 ATP and net 2 ATP. So whether it is anaerobic glucose oxidation or anaerobic glycolysis, this number will work, right? So good, very good. So quickly you have seen how you're going to revise the glycolysis, PDH complex, TCA cycle, and the energetics I have discussed, along with the glucokinase, hexokinase, and PFK1 activity, right? Now coming to next aspect of it, and I promised you that I'll be integrating various pathways also. So let's talk about the gluconeogenesis, right? So gluconeogenesis occurs in opposite condition in fasting or starvation. Glycolysis was occurring in well-fed state, but gluconeogenesis is going to take place in starvation or fasting state. And I said that majority of the steps of the glycolysis will be used in the reverse direction. See, there are many substrates which can produce you glucose: pyruvate, lactate, glycerol, propionyl-CoA, various amino acids. I am just trying to explain you how the pyruvate is making you glucose. See, it can go to the reverse order and make glucose, but you see in the glycolysis, 3 reactions are irreversible: pyruvate kinase, then PFK1 and glucokinase. All other reactions are reversible. So all reversible equations will be used, but irreversible equation need to be bypassed in gluconeogenesis. So pyruvate kinase needs shunting, PFK1 needs shunting, glucokinase needs shunting. These two shuntings are very easy, single enzyme catalyzed. But when we talk about pyruvase kinase shunting, it's a multi-step process. So pyruvate need to be processed properly to get converted to phosphoenol pyruvate, and after that there is no tension. All reversible enzymes will be used in the reverse direction, and fructose 1,6-bisphosphate will be formed. And once this is formed, you have exclusive enzyme of gluconeogenesis, that is 1,6-bisphosphatase enzyme, which will be countering the action of phosphofructokinase 1, and glucokinase counter will be glucose 6-phosphatase enzyme. Glucose 6-phosphatase enzyme, bachcho. And this is endoplasmic reticulum lumen enzyme. And this is the enzyme which is needed not only for gluconeogenesis but also for terminal step of liver glycogenolysis. So you must remember, this is such a cute enzyme which is very important to produce free glucose, not only from gluconeogenesis, also from liver glycogenolysis, right? So this is endoplasmic reticulum enzyme. Now coming back to that pyruvate kinase, I said multiple steps will be required. So let's talk about this pyruvate kinase shunting. So to save some time, I have already written the skeleton of this route by which pyruvate kinase is being shunted, and you can see the pyruvate is going to the mitochondria matrix. Pyruvate coming from alanine transamination going to mitochondria matrix. There it is undergoing carboxylation to give you 4-carbon oxaloacetate, and this enzyme is very important gluconeogenic enzyme, which is pyruvate carboxylase enzyme. And like any other carboxylase, it needs ATP, biotin, so all that tamjham will be required. Now this pyruvate will be converted to oxaloacetate. You know, it's an intermediate of TCA cycle. In fact, they call it as a second substrate. And there you see a reversible equation in the TCA cycle is there, which can convert malate to oxaloacetate, oxaloacetate to malate. So during gluconeogenesis, the malate dehydrogenase of TCA cycle is taking the reverse order, making you malate, and this malate comes out in the cytosol. This is cytosol of gluconeogenic cell. And there in the cytosol, again we have cytosolic malate dehydrogenase. This malate dehydrogenase converts malate to oxaloacetate, which is decarboxylated in presence of PEP carboxykinase enzyme. PEP carboxykinase enzyme, this is also very important gluconeogenic enzyme, and this is going to decarboxylate oxaloacetate, making you PEP. So this hassle of pyruvate kinase irreversibility is well taken by various enzymes here. You can see, if anybody asks you if the pyruvate is making you glucose, which TCA enzyme you think is very important, you have to say it is malate dehydrogenase enzyme, right? You may ask me one question, why oxaloacetate doesn't come out directly rather than coming via malate? The answer is very simple. Oxaloacetate cannot come out because it is having no transporter, it is not permeable. We have this malate transporter on the inner membrane, so malate very well can come out. So God has created mechanism like oxaloacetate will be converted to malate and will come out. So this is a malate transporter. It is not shuttle, right? That's a different thing altogether. It's a malate transporter which will bring the malate outside, and from there the PEP will be produced. Then you understand you have to use the enzyme in the reverse order, the enolase enzyme, the mutase enzyme, the PG kinase, everything will be used up, like, and glucose will be produced in liver, in kidney, in GIT cells in a minor quantity. Liver is the predominant organ, but don't underestimate the role of kidney in the process of gluconeogenesis. So in nutshell, this is gluconeogenesis. I have told you how the pyruvate is going to make you glucose. So understand the concept. Rest of it you can always build it, right? So you must know the glycolytic route, reversibility, irreversibility, then the shunting of irreversible reactions for the process of gluconeogenesis, right? And this is Cori cycle. Very important question to you every time. Cori cycle is nothing but it's a glucose lactate cycle. Glucose lactate cycle. In this, we talk about exercising muscle where anaerobic glycolysis is producing lactate. This lactate comes to the blood by diffusion, and that lactate goes to the liver for gluconeogenesis. Yes, lactate also can produce you glucose by gluconeogenesis. Only the thing is, like, lactate need to get converted to pyruvate first by LDH enzyme, and then pyruvate will be making you glucose. So glucose will be produced, which will be dumped to the blood, which will be utilized by the muscle. So it is like completing a full circle, and this is called Cori cycle, against the name of that scientist who discovered it, described it, right? So it's called glucose lactate cycle. So we have anaerobic glycolysis at one end and we have gluconeogenesis at another end. This is going to be a very, very important question to you every time, right? So जब ग्लूकोनियोजेनेसिस की बात की, मैंने कहा इसे भी छेड़ दूं. दिस इज कॉल्ड ग्लूकोज लैक्टेट साइकिल. एक और होता है काहिल साइकिल, राइट? उस पर मैं अभी नहीं जा रही डिटेल में. काहिल साइकिल होता है विच इज कॉल्ड ग्लूकोज एलानिन साइकिल. वहाँ भी हम ग्लूकोनियोजेनेसिस की बात करते हैं, राइट? बट हम वहाँ मसल प्रोटीन ब्रेकडाउन की बात करते हैं, राइट? सो दैट इज सीन इन स्टार्वेशन, काहिल साइकिल. ये ग्लूकोज लैक्टेट साइकिल तो हम बात करते हैं एनारोबिक ग्लाइकोलिसिस में, ठीक है? और राइट, जहाँ लैक्टेट प्रोड्यूस होता है. वेरी गुड बच्चों, फास्ट 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 आपको पूरा ओवरव्यू में बताकर जाने वाली हूँ कार्बोहाइड्रेट मेटाबॉलिज्म में. कमिंग टू द नेक्स्ट पाथवे which is very close to my heart, and that pathway is the galactose metabolism, right? So galactose we consume in our diet. Let me choose some better color pen here, right? Which color you want? All right, so galactose, this is converted in the liver to galactose 1-phosphate, glucose 1-phosphate, and then glucose 6-phosphate. That's it. This is the pathway of galactose metabolism. Ultimately, glucose 1-phosphate is converted to glucose 6-phosphate by phosphoglucomutase enzyme, and this glucose 6-phosphate will be either utilized in the glycolysis or will be stored in the form of glycogen, because glucose 6-phosphate can do anything in the cell, right? So this is how the galactose is ultimately going to enter in the glycolytic pathway. But what is very important for you to know that number one, the UDP glucose drops the glucose here and takes away the galactose, kidnaps the galactose out of this mainstream pathway. So this comes out and they are epimerized by 4-epimerase enzyme, right? And this is going to be galactokinase enzyme, bachcho, galactokinase, and this is going to be galactose 1-phosphate uridyl transferase enzyme. Never forget this name. You can call it as Gali-put enzyme for memory, right? Because it's a very important enzyme. I'll tell you why. It's galactose 1-phosphate uridyl transferase enzyme. So this is how the galactose metabolism takes place. Why we talk so much about galactose metabolism in the liver? Because any error in the pathway like galactokinase deficiency results in no utilization of galactose in the galactose metabolism. That time this galactose is high in the blood, goes to the lens, and aldol reductase, aldose reductase enzyme converts it to galactitol, lens of the eye, cataract in the eye. So benign galactosemia, this condition is called benign galactosemia, and baby will have cataract. It is not that benign. Oil drop cataract, baby will have cataract. But what is more dangerous is Galliput enzyme deficiency, which is the commonest galactosemia, and this was the galactosemia which was first described. It's called classical galactosemia. Galactosemia, where baby is having more than cataract. कैटरैक्ट तो है ही, बेबी के पास और भी बहुत सारी प्रॉब्लम्स हैं, मेंटल रिटार्डेशन है, कैटरैक्ट है, जॉन्डिस भी हो रखा है, लिवर एनलार्जमेंट भी हो रखा है, किडनी भी इन्वॉल्व है. This is the typical presentation of classical galactosemia, where mental retardation due to repeated hypoglycemia, right? Hepatomegaly, mental retardation due to repeated hypoglycemia. I'll tell you the reason for hypoglycemia. And also there is a organomegaly. I have told you, mental retardation, and then you have cataract. Yes, cataract, oil drop cataract. These are triad of manifestation in classical galactosemia, so you must know that. And as far as hypoglycemia, you are interested to know why this baby is going to have hypoglycemia. The answer is very simple. The galactose 1-phosphate is going to inhibit one enzyme which is crucial to break the glycogen and release the glucose in the blood, and that is rate limiting enzyme of glycogen phosphorylase. This has got many dushman. Galactose 1-phosphate is one of them. This is allosteric inhibitor of glycogen phosphorylase, so there is no glycogenolysis, no blood glucose, so baby gets repeated hypoglycemic attack, which affects the brain development, and baby develops mental retardation, right? So this is regarding the galactose pathway. You must know, I'm just like touching these important pathways. And coming to fructose metabolism, fructose we consume in diet, fructose converted to fructose 1-phosphate in liver. I'm talking only about liver. Fructose 1-phosphate, and this fructose 1-phosphate is immediately going to break into two. One is DHAP, another is glyceraldehyde. That's it. DHAP is utilized in the glycolysis. It will enter in the glycolytic route, and glyceraldehyde will say मुझे भी जाना है glycolysis में, but as such it cannot go, so it has to get converted to glyceraldehyde 3-phosphate. अब आप कहोगे कैसे होगा चेंज, तो ट्रायोकाइनेज है ना, ट्रायोकाइनेज 1 ATP लगाएगा उसका फॉस्फेट, राइट? And this enzyme is fructokinase enzyme using ATP. This enzyme is aldolase B enzyme, breaking fructose 1-phosphate. Everything is important for you, right? So this is how it is. All right. So, and yes, again the problem, the benign fructosuria, fructokinase deficiency, aldolase B deficiency, hereditary fructose intolerance. You need to read all these problems, you know, the fructokinase, the problem of fructokinase enzyme deficiency, and ये सच में benign है. The baby will be growing totally normally, no cataract, nothing, because cataract बनाने वाला enzyme जो है वो कौन सा है? Aldose reductase. It will act on aldose, not on fructose. Fructose is a keto sugar. So it's a fructokinase deficiency where fructose is high, and it's a completely benign condition. It's like will be detected accidentally by positive finding the positive Benedict test. And hereditary fructose intolerance, so very dangerous. Organomegaly, hypoglycemia, क्योंकि ये भी दुश्मन है किसका? ग्लाइकोजन फॉस्फोरिलेज का. So there will not be any glycogenolysis, hypoglycemia, mental retardation. Those problems will be there, right? So organomegaly, mental retardation sort of issues are there, and many other problems are there. For that detail of discussion on these cases, you need to watch my different video, which I'll be bringing shortly. All right, so this is regarding galactose and fructose metabolism. So what we have covered till now, we have covered the glycolysis, the PDH complex, the TCA cycle. We have seen how the galactose is ultimately going to glycolysis and the disorder associated with that. Fructose, how it is going to enter the glycolysis and the disorder associated with that, though not in big detail, but I have touched these topics, right? And yes, and in TCA cycle, when we are discussing, just to add 1 or 2 lines here, we have cataplerotic and anaplerotic reaction. एनाप्लेरोटिक इज फिलिंग अप रिएक्शन बच्चों, फिलिंग अप, ये फिल करते हैं टीसीए इंटरमीडिएट को, एंड कैटाप्लेरोटिक दे आर एम्प्टिंग रिएक्शन, वो खाली करते हैं टीसीए इंटरमीडिएट्स के एम्प्टिंग रिएक्शंस। आप देख सकते हो दिस इज टीसीए साइकिल इन ब्लैक कलर एरोज। यू कैन सी सिट्रेट मूविंग आउट, टहलने जा रहा है भाई, जा रहा हूँ बाहर, बहुत ज्यादा हूँ। इफ सिट्रेट इज एक्सेसिव इट विल गो एंड प्रोड्यूस यू फैटी एसिड, इट विल लीव द टीसीए साइकिल, गो आउट ऑफ द टीसीए साइकिल. This is a cataplerotic reaction because it is extracting the TCA intermediate outside. Cataplerotic reaction बच्चों, cataplerotic reaction. You can see you see oxaloacetate moving away in the form of aspartate, not in a mode of running the TCA cycle. Right, oxaloacetate moving away to form your phosphoenol pyruvate, then going to gluconeogenesis. I was explaining you some time back. So this is all cataplerotic reaction. Pyruvate converted to oxaloacetate by carboxylation. It is a pyruvate carboxylase enzyme. क्या होगा ये, कैटा या एना? Pyruvate giving you TCA intermediate, oxaloacetate. So it's an anaplerotic reaction. In fact, this is the most important human anaplerotic reaction, right? Then we have valine, isoleucine, methionine, not chain fatty acid. They all are going to produce you, provide you succinate CoA. So those reactions which are providing TCA intermediates are called succinate CoA. And you know, it can go away also in the heme synthesis because it's a precursor of heme biosynthesis. So this becomes a cataplerotic reaction. This is an anaplerotic reaction. So there must be a fine balance between cataplerotic and various anaplerotic reactions which will determine the actual rate of TCA cycle. So TCA cycle is having all the intermediates coming in and going away, depends upon their mood, right? So with this I stop this video, and in the next video I'll be talking about the rest other pathways. I hope you have enjoyed the session. If you like this video, you kindly share with your friends to spread the knowledge, and subscribe my channel for further such quick videos which will give you an overview of the metabolism, will make your life easy.